Have you ever wondered how scientists can use jellyfish DNA to make pigs glow, or how watermelons can be grown without seeds? It's because of biotechnology. Biotechnology is when scientists use living systems to make or modify products, and it is usually done to increase the quality of life or for scientific discovery. Biotechnology can be split into four main branches: genetic engineering, cloning, stem cell research, and synthesis of small molecules. Let's talk about genetic engineering first. Genetic engineering is modifying an organism's genetic code in order to give them different characteristics. This starts with gel electrophoresis, which works with restriction enzymes to cut DNA into fragments. This allows the original strand to merge with a new strand of foreign DNA. After this happens, the new strand of DNA from the donating organism is merged, usually by injection, into the plasmid area of the receiving organism. The enzyme DNA ligase helps facilitate this process by fusing the two DNA strands. This new, hybridized DNA strand is called recombinant DNA. It can create recombinant bacteria or hybrid organisms. These organisms, or microorganisms, will have both its own traits as well as the DNA traits from the foreign organism. This is how scientists make glow-in-the-dark pigs, as well as genetically modified foods like corn and seedless watermelons. The second branch of biotechnology is cloning, the genetic copying of an organism or its components. This means that scientists can either clone the entire organism or parts of its genetic makeup. When either gene cloning or DNA cloning is done on an organism, polymerase chain reaction, or PCR, is used. PCR is very similar to the genetic engineering process. The first thing that happens during PCR is the first DNA strand is split using gel electrophoresis and restriction enzymes. Next, the DNA is attached to another strand using DNA ligase and moved to a host cell. When the combined DNA strands are accepted by the cell creates many copies of it, completing PCR and the DNA cloning process. In organism cloning, the DNA is removed from the organism being cloned and added to an egg cell of another organism. This allows the DNA of the organism being cloned to develop. Once the DNA and the egg cell finish developing, a new organism with the exact same genetic makeup of the organism being cloned is created. Stem cells are cells in the body that have an extreme regenerative ability. The two main types of stem cells used by scientists are embryonic stem cells and adult stem cells. The main difference between the two is their ability to differentiate into different cells. Researchers are trying to use stem cells to treat diseases as well as heal damaged cells and tissues. While there is a lot of opportunity to use stem cells to benefit life, scientists are still learning about them and trying to figure out how they can be manipulated. The fourth and final branch of biotechnology is small molecule synthesis. Depending on the type of molecule, scientists use chemical reactions to combine them with other small molecules. For example, to make the drug aspirin, scientists create reaction in cornstarch, its main ingredient, to combine it with other ingredients like cellulose to create the drug. This is how most pharmaceutical drugs like Advil and Tylenol are made. Scientists can use biotechnology as a gateway for not just improving the quality of the lives of humans, but also saving the lives of humans. It is imperative that we continue learning more about DNA and genes so that biotechnology can continue in the future.